friends, welcome to my channel Easy Life Science. Here I am talking about some important questions, their answers with detailed explanations for SET exam subject Life Science part 8. So please subscribe my channel Easy Life Science, like my video, share it to your friends and press the bell icon so you will get the notification of my newly uploaded video. So let's start. The first question, the question is about a diffusion across a lipid bilayer. Choose the correct order of a molecule according to their ability to diffuse across a lipid bilayer. The large molecules, polar molecules and ions cannot easily pass through a cell membrane. Large molecules such as glucose, amino acids and proteins are too big to fit through the phospholipid bilayer. Because the cell membrane is a semi-permeable, only small uncharged substances like CO2 and O2 can easily diffuse across it. The CO2 across the cell membrane through a diffusion, glucose cross the cell membrane through a carrier proteins, H plus ions can cross the cell membrane through a protein channels, oxygen molecule can cross the cell membrane through a diffusion, auto molecule it can transfer through a diffusion by using aquaporins. The RNA molecules are polar because of negatively charged phosphate groups along a sugar, so they can cross a cell membrane very slowly. Glucose molecules are too big to diffuse through a plasma membrane. They are moved across the cell membrane through ion gated channels. So the gas molecule will diffuse fastly, so the CO2 will move across a cell membrane fastly. Then the water molecule, then a glucose and then the RNA. This is a sequence: CO2, H2O, glucose and RNA. So option A is the answer of this question. Then next question. 1 mole of a native protein upon a N-terminal analysis yielded 1 mole of each of aspartic acid and valine. Therefore, the protein in its native state exists as. Here, the one protein is degraded from a N-terminal. It is with 2 amino acids, and these 2 amino acids are different. So, it is a heterodimer. So, answer option C is the answer of this question. It is a heterodimer. Then, moving to the next question. The prostatic groups or cofactors involved in both 1 electron and 2 electron transfer in a mitochondrial electron transport chain are. Here a question is asked on the about electron transports. The coenzyme Q and flavin mononucleotides, these two are carriers which are present in a electron transport chain, and they can transfer 1 electron or 2 electrons. So answer of this question is option D. Moving to the next question. Indefinite stamen is a characteristic feature of which of the following plant family? The indefinite stamen, it is a characteristic of a family Malvaceae. The Malvaceae family show a indefinite stamen with united filaments in a tube. This condition is called as a monoadalpous. The family Cruciferae shows 6 stamens in a tetradynamous condition in which 4 stamens are longer than other 2. The family Solanaceae shows 5 stamens in a epipetalous condition, means stamens are attached to petals, whereas the family Gramineae shows 3 stamens in which there is a presence of free filaments. For this question, answer is a family Malvaceae. The family Malvaceae shows a indefinite stamen in a monoadalpous condition. So moving to the next question. Match the atoms in a group first with the most appropriate atom in a group second and choose a correct option. Integrins, microglial cells, toll-like receptor 7 and a dendritic cell. The first, that is a integrin. integrins. These are a transmembrane receptor that facilitate cell to cell and cell to extracellular matrix adhesion. Upon ligand binding, the integrins activates a signal transduction pathway that mediate cellular signals such as regulation of cell cycle, organization of intracellular cytoskeleton. The integrins works along with other proteins such as cadherin and selectins. Ligands for integrins include fibronectin, collagen, and laminin. Means integrins binds to a endo binds a cell to a endothelium. So the integrin matches with option 4. Then microglial cells. The microglial cells are the immunocells of a central nervous system and consequently plays an important role in a brain infection and inflammation. These cells are specialized macrophages found in a central nervous system. They remove damaged neurons and infections. The total glial cell population can be subdivided into four major groups. One microglia, astrocytes, oligodendrocyte, and Ng2 glia. Microglia in a culture secretes a large amount of hydrogen peroxide and nitric oxide in a process known as a respiratory burst. Both of these chemicals, that is hydrogen peroxide and nitric oxide, due to a respiratory burst, can directly damage the cell and leads to a neuronal cell death. So microglial cells are phagocytic in nature, found in a neural tissue. So they can match with the first in a group second. Then a toll-like receptor 7. The toll-like receptor are a class of proteins that play a key role in a innate immune system. They are single pass membrane spanning receptors usually expressed on a sentinel cells such as macrophages and dendritic cells. That cell recognize structurally conserved molecules derived from microbes. The toll-like receptor are a class of pattern recognition receptor, that is a PRRS, and these pattern recognition receptors that initiate the innate immune response. So the TLR7, that is a toll-like receptor, matches with a recognition of a single stranded RNA, that means a third. Then the dendritic cell are nothing but a antigen presenting cell, or these are a professional antigen presenting cell. These show a processed antigen to the CD4 plus cells or a T helper cell, and these are phagocytic in nature. So for a dendritic cell, they are a antigen presenting cell, they can process a antigen, so they match with a second. So this combination is in option B. Option B is the answer of this question. So moving to the next question. In natural condition, which of the following plant does not exhibit a anomalous secondary growth? The question is asked on anomalous secondary growth. The anomalous secondary growth or abnormal secondary growth is a growth in which fascicular cambium produces a xylem on inside and a phloem on outside. It is mainly observed in some monocots such as Dracaena, agave, yucca. These are the example of the plants Dracaena, yucca, and aloe or agave. In this type of growth, a series of cambia is formed outside the oldest phloem, which later forms patches of meristematic cell and conjunctive tissue. So here in this image, we can find for a secondary anomalous growth. Here is a transverse section of a bignonia stem and it is show anomalous secondary growth. This is a secondary xylem, secondary phloem. The importance of a secondary growth: old and non-functional tissue of plants are replaced with a new tissue. So all of the options, that is a B, C and D, aloe, yucca and daciana, these plants show a anomalous secondary growth, whereas the rice does not show a anomalous growth. So answer of this question is option A. Then moving to a next question. A typical angiosperm under a natural condition, primary meristem are usually established during. Now about a meristem, the meristem are classified as a primary meristem and a secondary meristem. A primary meristem is a form of a meristematic tissue that controls the growth of plant in terms of a length, and this is controlled by a root apical meristem or a shoot apical meristem. The secondary meristem is responsible for the increase in stem girth and thickness. The secondary growth is controlled by two lateral meristems called fascicular cambium and a core cambium. Proliferation of a cell leads to formation of an embryo in which two distinct meristem begins to form. So the formation of meristematic tissue or a meristem is occurs during a embryogenesis. The embryogenesis is a process in which there is a formation of embryo. So for this question, answer is a embryogenesis. In a embryogenesis, in a angiosperm, there is a primary meristem formation. Then moving to the next question, 2-methoxy 3,6-dichlorobenzoic acid belong to which class of a plant growth regulator? This is a synthetic auxin. The 2-methoxy 3,6-dichlorobenzoic acid, it is a synthetic auxin. So option A is the answer of this question. Then moving to the next question. In a typical green plant, the first stable end product of a Calvin cycle is the substrate, that is ribulose 1,5-bisphosphate in presence of carbon dioxide through a enzyme Rubisco get converted into a first stable compound in a Calvin cycle is a 3-phosphoglyceric acid, and this 3-phosphoglyceric acid is a 3-carbon containing compound and 2 moles of this compound are formed. So the answer of this question is option D, that is the 3-phosphoglyceric acid. Then moving to the next question. Among the following, which best describes an organism that lives at the expense of other organism, harmful but usually not killing? Here read the question carefully. Harmful, not killing. In this question, the 4 options are given, which shows a association in between the organism and their partner. The first option, that is A, predator. The predator, it kills their prey and lives. But in a question there is asking about a not killing, so it is not an option or not an answer. Then a symbiotic association. The symbiotic association is the association in which both the partners get benefited. So this is also not an option or a correct answer. Then a prey. In a association, the prey is get harmed by their predator. So it is also not an answer. Last, that is a parasite. The parasites feeds upon their host and finally kills their host, and this association is killing their host, not killing their host, but it is living inside the host. So answer of this question is the option D. Then the next question, Oleogum resin, that is Asafoetida hing, is obtained from the cut surface of. The Asafoetida is an oleogum resin, that is the exudates of roots of Ferula asafoetida. In a medicine, the asafoetida is acting as sedative, analgesic, antispasmodic, and a diuretic. Asafoetida is also called as a hing spice. In a regional language, Marathi, it is known as a hing. It is used as a giving a flavors to a curries, meatballs, and a pickles. It is in acrid taste. It emits a strong anion-like odor because of its organic sulfur compounds. Asafoetida is also used as a digestive aid or a menstrual problem. So it is a product of roots of a plant Ferula asafoetida. So the answer of this question is option B, that is roots. Then the next question, Bacne disease or foolish seedling disease is caused by. It is caused by one fungi, that is Gibberellia fujicuri. It is a fungi, Gibberellia fujicuri. It is because of high secretion of hormone gibberellins. Here there is a formation of a false seeds, so it is known as a foolish seedling, and a causative agent for this is a fungi Gibberellia fujicuri. Then next question, group first, group second, and group third represents enzyme, product of enzymatic reaction, and a metabolic process respectively. So choose the correct combination from group first, group second, and group third. Options are given. So starting from group first, hexokinase. The hexokinase is an enzyme that phosphorylates hexoses. Hexoses are 6 carbon containing sugars. After activity of enzyme hexokinase on a hexoses, hexoses are converted into hexose phosphate. Glucose is a most important substrate for the hexokinases, and glucose 6 phosphate is the product of it. Hexokinases transfer an inorganic phosphate group from a ATP to a substrate. Hexokinases should not be confused with glucokinases. Glucokinases are a specific isoform of hexokinases. All hexokinases are capable of phosphorylating several hexose sugars, but glucokinase acts with a 50 times fold lower substrate affinity, and its main hexose sugar substrate is a glucose. So glucokinase has a lower affinity to the glucose. So hexokinases, these are an enzyme in a first step of a glycolysis, acts on a glucose and forms a glucose 6 phosphate. Glucose gets phosphorylated through the activity of hexokinase. ATP is hydrolyzed to a ADP and a inorganic phosphate. This phosphate is accepted by a glucose and forms a glucose 6 phosphate. So this is a reaction of a glycolysis. Then a fumarase. Fumarase or a fumarate hydratase is an enzyme that catalyzes the reversible hydration or a dehydration of a fumaric acid to a malic acid. And this is a reaction of a TCA cycle, fumarase TCA cycle. So the fumarase get converted into a malic acid or a malate in a TCA cycle. Then a third enzyme, PEP carboxylase, phosphoenol pyruvate carboxylase, also known as a PEP carboxylase. It is an enzyme in the family of carboxylases found in plants and some bacteria. This enzyme catalyzes the addition of bicarbonate to a phosphoenol pyruvate to form the 4 carbon containing compound oxaloacetate and a inorganic phosphate. This reaction is used for a carbon fixation in a CAM pathway. CAM pathway stands for a crustacean acid metabolism and also in a C4 pathway. So this occurs in a photosynthesis and its product is a oxaloacetic acid, PEP carboxylase, oxaloacetic acid and a photosynthesis. Then glycolate oxidase. Glycolate oxidizes glycolic acid to a glyoxylate or it can also oxidize glycolic acid into a oxalate. So glycolate oxidase hydrogen peroxide in a photorespiration. So this combination is in option B. So option B is the answer of this question. Then moving to the next question. Here also the two groups are there. Match the following in a correct combination between group first and a group second with reference to agent that interfere with a oxidative phosphorylation. In a group first, some electron transport chain inhibitors are listed, and in group second their activity or their mechanism is listed. You have to match it. The first, that is a cyanide. Cyanide is an inhibitor of a mitochondrial electron transport chain. It binds to a heme A3 prostatic group in a complex 4th, that is a cytochrome C oxidase. Toxicity of a cyanide is cessation of aerobic metabolism in a cell. After cyanide poisoning, the electron transport chain can no longer pumps the electron into the intermembrane phase. The pH of the intermembrane phase would increase, ATP synthesis would get stopped due to the cyanide. So cyanide inhibit the complex 4, that is inhibit the cytochrome oxidase. Then the antimycin A. Antimycin A inhibit the flow of electron from a cytochrome B to a cytochrome C1. It uncouples a oxidative phosphorylation in which ATP production is less. So the antimycin matches with the blocks electron transport from cytochrome B to a C1, Q to first. Then aroferritin. The aroferritin is naturally occurring antibiotic. It interacts with a beta subunit of a F1 domains in a ATP synthase enzyme. It means it inhibit a F1 domain or a F1 particle in a ATP synthase enzyme complex. So R with a second. Aroferritin to a inhibits a F1. Then a fourth 2,4-dinitrophenol. The DNP or a 2,4-dinitrophenol acts as a proton ionophore, and an agent dissipates the proton gradients across the mitochondrial membrane, collapsing the proton motive force that the cell uses to produce most of its ATP. Due to DNP, cell instead of producing ATP, the energy of a proton gradient is lost as a heat. So the DNP or a 2,4-dinitrophenol is an uncoupling agent in a ETC. It uncouples a phosphorylation from a electron transports. So this matches with the third. So this combination is in option first. So answer of this question is option A. Then moving to the next question. Here the some inhibitors of electron transport chain are listed. The cyanide, carbon monoxide. These inhibit complex 4, that is cytochrome oxidase, cyanide and carbon monoxide. Then the antimycin A, mixothiazole, rotenone, amytal, pyridine A. This prevents a transfer of an electron from APS carrier to a ubiquinone. Dichloromethyl or a DCMU. This compete with coenzymes to binding in a site in a photosystem second, so it inhibit a photosynthesis. Then inhibition of ATP synthase, arovatin oligomycin, DCD. These are inhibitors of ATP synthase complex in ATC. They binds or they inhibit F1 or F0 particles. Some uncouplers are listed here, that is DNP, FCCP, phenylamycin and thermogenin. Some inhibit exchange of ATP to ADP, the atractylosides. It inhibits adenine nucleotide translocase enzyme. So these are the enzymes. Sorry, these are the inhibitors in electron transport chain. Most of times questions are asked on the SET lifetime exam. So take a screenshot of it. Then next question, which of the following chemical is used for doubling of a chromosome numbers during a production of doubled haploids in a crop plant? Doubled haploids is a genotype formed when haploid cells undergo a chromosome doubling, and for this chromosome doubling colchicine treatment is used. This colchicine makes a doubled haploid. So answer of this question is option C. So thank you for watching my video. Please subscribe my channel Easy Life Science. Like my video, share it to your friends, and don't forget to press the bell icon so you will get the notification of my newly uploaded video. Thank you once again.